So, Sana, this is what we had done in the last class. Okay, we had done about the transcription process. Okay, see,
Yeah.
then we have started about the protein synthesis, just a little bit of explanation. Okay, so today what we are going to do is we are going to go on the genetic code, and then we are going to go into the process of translation. So you already know how the ribosomes will carry out translation, or how the ribosomes are attached. So let's start with the genetic code, and then we will go to the process of translation. Shall we start?
Yeah.
Okay. So translation is nothing but the other name of protein synthesis. Clear?
Mhm.
Okay. So let's go to genetic code. Now, we need to understand here is that from DNA we get mRNA. mRNA is messenger RNA. Now, whatever the information is in the form of a code in DNA, that is transferred to the mRNA, and it stores in the form of what we call as triplet codons. Okay, triplet codons. That means these codons are nothing but they are nitrogen bases. Okay, they are nitrogen bases, but they occur three together. Okay, they occur three together. So genetic code. All right. So how will you define the genetic code? It's a relationship between the sequence of amino acid in a protein chain or a polypeptide chain. Okay, sequence of amino acids. So suppose, for example, if the suppose this is a protein. Suppose this is a protein. So which amino acid? Oops, one second. Which amino acid is present here? We may have different amino acids. So this is a particular type of protein, and the sequence of the amino acids are also different. Okay. Now,
Yeah.
you know that we have 20 amino acids, and that 20 amino acids are shuffled in different ways to form the different type of proteins. So, genetic code means it tells us what will be the sequence of a protein. Okay, what will be? For example, valine is a amino acid. So its triplet codon will be G U G. Guanine, uracil, and guanine. These three nitrogen base represents valine. So if there is GUG in the RNA, it means that in the protein, what will be there? Valine amino acid will be there. This is clear or not?
Yeah.
And triplet codon, why? Because there are three numbers. See, guanine, uracil, guanine, GUG. Okay,
Mm-hmm.
so it's triplet codon. Codon is actually, yes. Any doubt?
No.
No. Codon is actually the information that is present in a DNA.
Mm-hmm.
Okay, that information is transferred to the RNA in the form of codon. So codon will store all the information. Okay, it will store all the information. Okay, and see this. So valine, like that. Suppose, for example, okay, if we have cysteine, then this will be UGU. Okay, so valine, cysteine, again valine, cysteine, valine, whatever. So this particular protein has this sequence. Another protein will have the same amino acid, but the sequence may differ. Here, valine and cysteine is there. There can be valine and thymine. Okay, tryptophan. Anything can be there. So when we are trying to look into the protein, how the polypeptide chains, how the amino acids are arranged, that is an important thing. Did you understand, Sana? Yes?
Yeah.
Yeah. So what happens? We may have many protein who may have valine, who may have cysteine. Okay, all this may be there in a protein. But what it differs is that these two are there in all the proteins. But the difference is that in which position they are placed. What is the sequence? In one amino acid, valine may be first, and in another amino acid, valine may be last. Okay, understood or not? So every protein will have a different sequence of amino acid. Okay, so every protein will have valine, cysteine. Okay, but what is the position of the amino acid there? That differs from one protein to the other. Have you understood this, Sana?
Yeah.
Yes, any doubt here? Because this is
No.
very important. You need to understand sequence of amino. We may have thousands of proteins. All thousands of proteins will have valine, cysteine, tryptophan. But in one amino acid, it may be valine, cysteine, tryptophan. Another amino acid, tryptophan, valine, cysteine, like this. That means amino acids are shuffled. Okay.
Mm-hmm.
Yes. Do you understand?
Yeah.
Okay. Each amino acid is represented by three letter words, which we call as the triplet codon. Okay, three letter words, which we call it as the triplet codon. Now, here, okay, GUG refers to valine, UGU refers to cysteine. Okay, so three triplet codons. So like this, what happens? Okay, so what happens maybe for example, let's see. And there's an amino acid phenylalanine. Phenylalanine. Phenylalanine is coded by UUU UUC. Okay. So whenever you see UUU, it always codes for the amino acid phenylalanine. When you code UUC, that also codes for phenylalanine. Okay. Understood
Yeah.
or not? Yes. So this is one amino acid. Phenylalanine is one amino acid, but it is coded by how many triplet codons? One or two?
Um, two.
Two. Okay, it's
Yeah.
coded by two. Like leucine. Leucine is an amino acid. It will be coded by UUA, UUG, CUU, CUC, CUA, CUG. How many triplet codons? It is coding. How many triplet codons will code for leucine?
Two.
Yes, how many?
Two.
All of these, yes or no?
Yeah.
Yeah, so how many are there?
Um, six.
Six. So it can happen that one amino acid can be coded by one triplet codon. It can also have that one amino acid is coded by six triplet codons. Did you see this?
Yeah.
Yes, understood or not?
Yeah.
Yeah. Clear or not, Sana? Yes.
Yeah. Understand.
Yes. Okay. So phenylalanine is coded by how many amino acids? Two amino acids. Leucine is coded by 6. Yes or no?
Yeah.
Yeah. So tell me, Sana, do you have any confusion at this point? See, if you understand this, if your concept on this is clear, the remaining section will be easy. But you
Mm-hmm.
need to clear your concept at this section. Tell me. I'm going a little bit slow. See, try to understand. If you have any doubt, speak up.
Mm-hmm.
Yes, tell me.
Um,
All
I
okay?
think, yeah, I think I understand.
Okay. All right. Okay. So we have many, you can say we have many genetic codes. Okay. So we have many amino acids. Okay. So in total, we have, okay. Now, when we talk about the genetic code, when we talk about all this, let's see here. We have 20 amino acid. Okay,
Mm-hmm.
20 amino acids, and we have 64 triplet codons. How many we have?
64.
64 triplet codons. Is this clear?
Yeah.
Yes. Okay. So let's see here. Well, okay. So phenylalanine, I told you, it's coded by how many amino acid? How many triplet codons? Two. Yes or no?
Yeah.
And okay. And then leucine, we have six. Yes or no? Yes,
Yeah.
leucine, we have six. Okay. All right. So here, when we are talking about 64 triplet codons, out of that 64 triplet codons, okay, Sana, you have done this chapter, right?
Yeah.
Yeah. So can you tell me, out of the 64 triplet codons,
Mm-hmm.
okay, out of the 64 triplet codons, how many are, how many will code for the different amino acids, and how many do not code for any different amino acids, so they are called as stop codons? Can you tell me, out
Um,
of the 64,
mm,
out of the
I,
64, think, out of the 64, 61 triplet codons will code for amino acids. 61, 62, 63, 64. So 3 are called as stop codons or nonsense codons. What are they called? Stop codons
mm-hmm.
or nonsense codons. That means, oops, that means these stop codons or nonsense codons do not code for any amino acids. Is this point clear?
Yeah.
Yes. Okay. So, whenever we are talking about all the amino acids out of that 64, okay, out of that 64, we have three stop codons, three stop codons, okay, within the 64. And we have two initiation codons. Initiation codons means these will initiate the process of protein synthesis. It will start the process of protein synthesis. Stop codon means it will stop the process of protein synthesis. Okay.
Yeah.
Yeah, because where are these where are these amino acids present? This is 5 prime, this is 3 prime. This is an mRNA. It's an mRNA. Okay, it's an mRNA. All right, then let's see here. First we will have AUG. Okay, AUG. Then we may have, let's see, CGU, like this. All right, CGU, and so on. Finally, we will have the three, we have any one of the three stop codons. UAA. Three stop codons are UAA. UAA, UAG, and UGA. UAG and UGA. These are the stop codons.
Mhm.
So now, if during the process of translation, the tRNA will read AUG, it will initiate the process of translation or protein synthesis. That means it will start. You have an mRNA strand, but in that mRNA strand, there is no AUG. That means whether how long that mRNA is, no AUG means it cannot initiate the process of translation. Okay, this is called as initiation codon. Whenever tRNA reads the amino or the triplet codon AUG, that means it has to start. Without AUG, that mRNA cannot start the process of protein synthesis. Initiation codon. Okay, initiation codon. So as initiation codon starts, goes on making amino acid. Now, the moment it reads UAA or UAG or UGA, anything, any one of this, that means this is called as the stop codon or termination codon. Stop codon. Stop codon. That means whenever a tRNA reads the stop codon, that means it has to stop the process of protein synthesis. It should terminate the process of protein synthesis.
Mhm.
Sana, do you understand this?
Yeah.
Yeah, any doubt?
No.
Okay.
Yeah.
Hmm. All right. Okay. So we have a start codon, we have a stop codon. So this is your start codon, and this is your stop codon. Okay. Now let's see here. Start codon AUG. Okay, AUG. All right. Codes for methionine and GUG, which is also an initiator codon, codes for valine. Okay, AUG, the start codon codes for which amino acid? Methionine. Okay, the other start codon, that is GUG, is also start codon. It codes for valine. Is this clear?
Yeah.
Yes, understood or not? Sana, are you understanding or not? Please tell me.
Yeah, I understand.
Mm. Okay. So out of this AUG and GUG, Sana, can you tell me which is the most common initiation codon? Both are initiator codon,
Mm-hmm.
but which is the most common initiation codon?
AUG.
AUG. Very good. AUG is the most common initiation codon. We very rarely have the GUG. Okay. Now, all 20 amino acids, each of them will be coded by different triplet codons. They will not be coded. Suppose AUG codes for methionine. So AUG also cannot code for valine. AUG for methionine, that means only for methionine. Okay, only for methionine. Is this clear?
Yeah.
Yes. So it's only for methionine. That means the 20 amino acids are there which are coded by different triplet codons. Okay, different triplet codons, but not same triplet codon. Okay, but not same triplet codon. Have you understood this? Yes. Any doubt?
No.
Okay. All right. Okay. So let's see this. Okay. So these codes that we have, all the codes, these are universal. These are what? Universal.
Mm-hmm.
Got it? That means if you are there, you will, if you say methionine, you will use AUG.
Mm-hmm.
If I'm here, if I say methionine, I will say AUG only. It's
Mm-hmm.
not like that in your place. It is said methionine is coded by different triplet codon. In my place, it is coded by different triplet codon. No, it is the same. It
Mm-hmm.
is the same. Clear?
Yeah.
Okay. All right. Okay. Understood or not? All right.
Yeah.
So these are some of the properties of the amino acid. Okay. These are some of
Mm-hmm.
the properties of the amino acid. Okay. All right. Then, um, okay. So what we will do now is that, one second. Okay. So since you have understood the triplet codons, okay, now what we will do is that we will jump to the process of translation. Translation is called as protein synthesis. Clear, Sana? Yes. Sana,
Yeah.
have you understood triplet codons? Yes. If you want
Yeah.
some time, you take time and go through. Okay, because I don't want you to just pile up triplet codon, because it's very important. Without triplet codon, you cannot understand protein synthesis or
Mm-hmm.
translation. Okay, understood or not?
Yeah.
Yeah. Okay. So let's see this. We'll do the process of protein synthesis. Okay. So translation or protein synthesis. Okay. Now, for protein synthesis, what are the requirements that we need? The first thing that we will need is ribosome. Ribosome are called as protein factories. Okay, ribosome are called as protein factories. So we know ribosomes are those structures where the process of protein synthesis will occur. Okay, so what happens? we have to keep the two subunits of ribosomes intact. Okay, two units of ribosome. So this is done by Mg2 plus. Magnesium ion will keep or attach the two subunits of ribosomes together so that protein synthesis can be completed. This is larger subunit. This is smaller subunit. Okay, all right. So let's see here. Okay, so now, okay, so now what we will do is that we will try to understand how the process of protein synthesis occurs. So we need ribosome. Secondly, we need the enzymes for protein synthesis. Enzymes for protein synthesis. Okay, enzymes for protein synthesis. Then we will see, let's see what is required. Okay, so we'll have also mRNA, tRNA. Okay, mRNA, tRNA. mRNA and tRNA. Okay,
Mhm.
all right. These are the requirements. Okay, then we will also need, I told you, enzymes here. Okay. One second, I'll just give you the name of the enzyme. Okay.
Mhm.
Amino acyl tRNA synthase. Okay. All right. So these are the requirements for protein synthesis. Okay. So let's see the process of protein synthesis. Okay.
Mhm.
Let's see the process of protein synthesis. One second. Okay. Larger subunit. All right. So TRM ribosome will have A site. It will have P site, and it will have E site. A site, P site, and E site. Ape. Okay, you can remember it as ape. Okay. All right. Then let's see this. Ape. Okay. So A site. Okay. A site is where the activation. Oops, sorry. One second. I'll write it properly. Site is where the activation activation of tRNA takes place. Okay. P is the site of peptidyl transferase, and E site is exit site. Okay.
Yeah.
So here the tRNA will be activated. Now, what happens? The tRNA comes with its anticodon. Okay. Now, here we have the mRNA. Suppose here what happens? We are reading. Okay, we are reading the first amino acid. Let's see here. Suppose what was the first amino acid initiation codon? I told you
AUG.
AUG. Yes, AUG. So this tRNA comes. Okay, it reads that AUG. So when tRNA reads AUG, what it will be?
Um,
U
U
A
A
C, right?
C. Yeah.
Yes. First of all, the tRNA and mRNA will form bond. After that,
Mhm.
they will transfer. If it is AUG here, this anticodon, this is the anticodon loop.
Mhm.
Okay. So A converts to U converts to A, and G converts to C. So Sana, do you have your textbook with you?
No, not right now. I kept it in school today.
Okay, 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 no problem. So, can you, yes. So, suppose, for example, if it is AUG, okay, so it'll code for what? UAC. Okay,
Mhm.
so let's see one thing here. Okay, so UAC codes for which amino acid? It codes for tyrosine, tyrosine codes for TRY. Clear?
Mhm.
Okay. So, so when we talk about TRYUAC, all right, so this is AUG here. Okay, so okay. So yeah, so we have a protein, we have an amino acid tyrosine here. So now this is over. So this amino acid will go to P site. Then after that, it will go to this particular tRNA, and then it will exit. So the tRNA will exit. Okay, tRNA exit. Clear?
Mhm.
Okay. Now, okay. So now UAC, the first amino acid will form. Now, like this, many tRNA will come and code for the different amino acids. Slowly, slowly, what happens, in this P site, there will be many amino acids which will be accumulating. So peptidal transferase, this enzyme, what it is going to do is that it is going to slowly, slowly form the bonds between the amino acid. Peptidal transferase, they form peptide bonds. Okay, they form peptide bonds. Okay, they'll form peptide bonds and then they'll form a protein like this. See? Can you see this?
Yeah.
Peptide bonds. Okay, they form peptide bonds to form protein chain or polypeptide chain. Is this process clear?
Yeah.
Yes or no?
Yeah.
Sana, tell me if this is the mRNA.
Mhm.
Okay, this is the mRNA. So in this mRNA, okay, in this mRNA, let's see here. This mRNA,
Mhm.
who will take the information from the mRNA? to form amino acids? Who will take
The
the information? Yes?
the ca I forgot.
Wow, what
It's
is it?
just the C.
tRNA.
Mm-hmm.
Yes or no? tRNA, this is mRNA. See, suppose this is mRNA. tRNA will come, take the information, and form the protein. Are
Okay.
you getting my point? Yes?
Yeah.
Okay.
Yeah.
All right. Okay. So once to the exit side, they will exit. Peptidal transferase, what happens? Only accumulating amino acid is not enough because we are forming protein. So they will form the peptide bonds. Okay. Peptidal transferase will form what bonds? Peptide bonds. Understood or not?
Yeah.
Okay, so once what happened, the formation, the peptide bond will continue to form like this. It will continue to form.
Mhm.
Okay, the moment, when will the protein synthesis stop? Can you tell me when the protein
Um,
synthesis will stop? When they will read what codon?
five.
When they will read which codon? Stop codon or
Oh,
start codon?
um, stop codon.
Stop codon, right? Yes or no?
Yeah.
So
Yeah.
stop codons are either UAA, UGA, and UAG. Clear?
Yeah.
Mm. Understood or not?
Yeah.
The moment tRNA reads any three of these are stop codons.
Mhm.
Okay, so when the tRNA reads any three of this, so it means the tRNA means it understand it. Okay, I don't have to form any more amino acid. I'm free to go. That means this is the termination. The presence of the stop codon indicates the termination of translation.
Yeah.
To understand? Yes.
Yeah.
Yes, Sana. Yes.
Yes.
Okay. See, I want you to be very much expert in this particular topic. It's very easy, but you need to
Mhm.
understand few more important things. Yes or no?
Yeah.
Okay. Okay. Understood. Any doubt here?
No.
Clear?
Yes.
Translation is clear? Yes.
Yeah.
Without the activation of tRNA in the A site, please look into the board. I'm explaining. Without
Mhm.
the activation of the tRNA in the A site, tRNA cannot take the information from the mRNA. Clear?
Yeah.
Okay, so there has to be activation of tRNA.
Mhm.
Okay, okay. This is clear, right? Any doubt here?
No.
Okay. Okay. So this is the process of translation. So translation helps to form what amino helps to form protein. Yes or no? The process of protein formation or protein synthesis is called as translation, or it is called as simply as called as protein synthesis. Clear or not? Okay. Now see here. See, Sana. This is the larger subunit of ribosome. When we were doing cell biology, we have studied no structure of ribosome.
Yeah.
This is the larger subunit. Okay. This is the smaller, yeah, subunit. Now, the two subunits, Sana, they remain together by an ion called magnesium ion. but they remain together only for protein synthesis. When the protein synthesis is over, the two subunits separate out. That means ribosomes, two subunit do not remain together always. They come together only during the process of protein synthesis. This point is clear?
Yeah.
Hmm. Okay, understood now?
Yeah.
Hmm. Okay, so this is all about the process of protein synthesis. Okay, any doubt at this point to tell me? Protein synthesis, in the last class we had done translation. Any doubt?
No.
No, clear? Okay,
Yeah.
so summer we have done polypeptides, we have protein, amino acids, all we have done. Okay, uh, enzyme activity, I think we have done. Yes or no?
Yeah.
I don't remember. Yes, we have done enzyme. Yes, space sequences. Okay, one second. Let me see. Okay, all right. So, can you, Asana, if you have any idea, can one second. I will just check 1 second if we have done enzyme. Do you remember we have done enzyme?
Yeah, I think so.
Let me see from the first. Functions of proteins we have done. Ah, yes, we have done enzyme. See?
Yeah.
Yeah, we have done enzyme. Correct. We have done enzyme. Yes or no? Enzyme
Yeah.
functions and all. Okay. So, can you tell me, Sana, after that, which portion is done for you?
Um, in school or
Yeah, structure of DNA,
yeah,
RNA gene
we're
replication.
done with unit 2. We're
Huh.
starting topic 3.
Topic 3, right? Okay, so
Yeah.
photosynthesis you have done?
Yeah.
Okay,
In topic 2.
photosynthesis. Yes, yes, sorry. Sorry, Sana. What did you say?
Photosynthesis in topic 2.
In topic 2, yes.
I think so. I don't.
Hmm.
Mhm.
Photosynthesis and respiration. Topic 3 is genetics, right?
Yeah.
Yeah. So photosynthesis. So shall we do photosynthesis today?
Okay.
All right. Okay. Okay. Okay, we'll do photosynthesis. Clear?
Mm-hmm.
Okay. So, Sana, what do you know about photosynthesis?
It's the process in which plants
Hmm.
obtain
Up-
ATP.
ATP, no, they make their food, right?
Oh,
Yes or
yeah.
no?
Yeah.
It is a type of physico-chemical reaction. It's a biological reaction. Okay,
Mm-hmm.
so in photosynthesis is performed by the plants.
Yeah.
Okay, photosynthesis. 1 second. Performed by the plants. So in photosynthesis, what happens? light energy is converted to chemical energy. Light energy is converted to what? Chemical energy. Do you understand this?
Yeah.
Yes. So that means plants are able to take the light and convert the light into chemical energy. So this chemical energy is basically the formation or production of starch. Clear?
Yeah.
Which is the food of the plant. Okay. Photosynthesis. So in photosynthesis, if we talk about photosynthesis, so here, okay, we have, there were different scientists, there were different scientists who worked for the who worked for to study the process of photosynthesis. Various scientists were there. Okay, various scientists were there. So now what we are going to do is that we are going to start, though we're going to start understanding the chemical processes that is going on inside the plants. Okay,
Yeah.
so let's see here. Now the question is, light energy converted to chemical energy, who does that? So here in the plants there are certain pigments called as chlorophyll. Okay, certain pigments which is called as what? Chlorophyll.
Mhm.
So these chlorophyll are able to trap the light energy and convert the light energy into chemical energy. Clear on this?
Yeah.
Okay. So now, uh, now the range of wavelength of visible light is 400 to 700 nanometer. Okay, so here what happens? So this visible light between 400 to 700 nanometer. Okay, this is the light between which what happened, the absorption of chlorophyll will take place. That means chlorophyll will absorb the light waves between 400 to 700 nanometer only. Okay, 400 to 700 nanometer only. 400 is blue light, 700 is red light. Okay, blue light
Yeah.
and light. Clear? So that means I can say that photosynthesis is, one second. Photosynthesis is most active in blue light as well as in red light. And this was proved by T.W. Engelmann. He said that, okay, in 400 nanometer and in 700 nanometer, the rate of photosynthesis is highest. So these are called as photosynthetically active radiation. Photosynthetically active radiation. Is this clear?
Yeah.
Okay. Then next, let's see here. Okay, so Sana, can you tell me what do you understand by photosynthetically active radiation?
Mm, there's a
Yes.
active radiation.
Yes. What is it?
um, I'm not sure.
Yes, photosynthetically active radiation. That means it is in the blue light and the red light we have maximum rate of photosynthesis. Clear?
Yeah.
Yeah. So in blue light, that is 400 nanometer, and red light, which is 700 nanometer. Okay, in this two red light range, red, sorry, first is blue and red light. Okay, blue and red light, what happened? Here, the rate of photosynthesis is more, or it is maximum. Did you understand this, Sana?
Yeah.
Okay. So we have 400 and 700 nanometer. So 400 nanometer is blue light. 700 nanometer is red light. Okay. All right. So between this, at the two extremes, we have a very high rate of photosynthesis. But here the photosynthesis rate is low. Please remember, it does not stop. It is low. The reason why, because at 400 and 700, 400 and 700, chlorophyll is operational. Chlorophyll becomes the main pigment to absorb 400 and at 700 nanometer wavelength. But in between this, chlorophyll does not absorb. Rather, carotenoids absorb. Okay, carotenoids will absorb. So what happens? Carotenoids will not allow the process of photosynthesis to be stopped. Okay, it will not allow the process of photosynthesis to stop. Rather, it will go very slowly. So it's a low rate of photosynthesis. This point, Sana, is clear to you?
Yeah.
Okay. Okay. All right. All right. Okay. So suppose, for example, if the rate of photosynthesis decreases, if the rate of photosynthesis will decrease, okay, if it decrease after 700 nanometer, okay, see, suppose for example, after 700 nanometer we apply more light. Okay, so if the light is available after 700 nanometer, then what happens? Here, the rate of photosynthesis will become low and even stop. So this region is called as red drop. Okay, it is called as red drop. That means beyond red light, there is no photosynthesis. Now, how do I how do I bring about photosynthesis here? So in red drop, okay, so we don't have any, because red light is not there, so there is a steep drop in photosynthesis. So again I supply with light, red light. So when I supply with red light, what happens? Again the rate of photosynthesis will become high. That means for photosynthesis to continue, we should have blue light and definitely we should have the red light. So here red light was stopped, red drop. So when the red light was Stop. It is called as red drop, where the rate of photosynthesis decreased. Okay, where the rate of photosynthesis decreased. So what happened here is that now, okay, so it's called as a red drop. Okay, it's called as red drop. So what happens? There was a scientist. His name was Emerson.
Mm-hmm.
Okay, there was a scientist. His name was Emerson. So Emerson said that, okay, he said that in this case, in such case where the red light drops, if we supply the photosynthesis with more red light, then again the rate of photosynthesis will be high. Okay, so it is called as Emerson's enhancement reaction. Okay, Emerson's enhancement reaction. That means the red light is less, so we are supplying the photosynthetic apparatus with more red light so that it can continue the process of photosynthesis. Clear?
Yeah.
Okay. Okay. All right. So now when we talk about photosynthesis, okay, so photosynthesis, two things we need to understand here is that first thing is absorption, absorption spectrum. Okay, and we have action spectrum, absorption spectrum and action spectrum. So Sana, can you tell me in absorption spectrum in which light the chlorophyll will absorb more light?
Blue.
Blue and
Blue.
red, yes or no?
Yeah.
Yes. So absorption spectrum, that means it is in that the plant will absorb blue plus red light. Action spectrum, action spectrum is that in blue and red light only, but here we will, what we will see, action spectrum. That means in blue and red light we see an increased rate of photosynthesis. Is this point clear? Yes.
Yeah.
Hmm. So let's see here. Okay, the process of photosynthesis, absorption spectrum and action spectrum. Okay, so let me see here what are the things here. Okay, now, okay, so what happens now? We should understand that which molecule actually absorbs the light. So let's start here. Light comes in the form of discrete units, discrete units called photons. Okay, so light is absorbed in the form of discrete unit called photons. These photons are the one that the chlorophyll molecule will absorb. So what happens? In every plant cell, there are structures called chloroplast. In the chloroplast, okay, let's see here. In the chloroplast, one second. Yeah, okay. So we have chlorophyll A. Okay, all right. Chlorophyll A. Chlorophyll A is called as reaction spectrum. Okay. Now What are these? All these are called as antenna.
Yeah.
Antenna molecules. Or I can say these are accessory pigments. Okay, accessory pigments. Is this point clear?
Mm-hmm.
Okay. Now what happens? Now light energy falls. Oh, sorry. Light energy falls on these molecules. Okay, not on chlorophyll A directly. It falls in the accessory pigments or the antenna molecules. Once the light falls on the accessory pigments and antenna molecules, these starts to accelerate energy from one antenna molecule to the other. And ultimately, they will reach into chlorophyll A. Okay, chlorophyll A. And the electron in the outermost shell of chlorophyll, what happens? Because it is getting energy, it gets excited, it gets accelerated, and it moves to a higher energy level.
Mm-hmm.
Okay, moves to a higher energy level. This entire system is actually trapping light. Try to understand this. This entire system is actually trapping the light. Yes or no?
Yeah.
Yeah. So it is called as light harvesting complex. What is this called?
Harvesting complex.
Complex. Or we call it as the LHC. Okay, LHC. Now, suppose for now, see, okay, this did you understand?
Yeah.
Yeah, okay. So, in the plants, when we talk about in the plants, plants have two photosystem. Okay, plants have how many photosystem? Two. So, we have photosystem 1 and we have photosystem 2. Photosystem 1 and photosystem 2. Clear?
Yeah.
Okay. So, what is photosystem? It's a system of pigments. Okay, it's a system of pigments which helps to produce more sugar. Again, I'm repeating, it is a system of pigments which helps to produce more sugar. Okay, so photosystem 1 is active at 700 nanometer. Photosystem 2 is active at 680 nanometer. Okay, these 2 points are clear.
Yeah.
Okay, so the photosynthesis in plants is between this 680 to 700 nanometer. Clear? Photosystem 1 and photosystem 2. So
Mm-hmm.
the light harvesting complex, just now which I explained, of photosystem 1 or 700 is called as the LHC complex 1. LHC complex 1. And the one here, this is LHC complex 2. Okay, light harvesting complex 1 and light harvesting complex 2. Light harvesting complex belonging to photosystem 1 operating at 700 nanometer is called as LHC complex 1, and the other one operating at 680 nanometer is called as LHC complex 2. Till here, these are understood, Sana. Please have a look. If you've not understood anything, you tell me.
Yeah.
Yes, cleared out.
1 second.
Sorry.
1 second. Let me just.
Yeah, yeah, sure, sure, no problem.
Yeah. I'm okay.
Okay, all right. So, Sana, have you studied photosynthesis?
Yeah.
Yeah. What and all did you study in photosynthesis in school? Could you tell me?
Like the transportation of the plant. Like we learned that in HL.
Sorry?
We learned like transportation in like xylem and phloem
Hmm.
in HL.
Then, sorry?
In HL, like in our higher level
Okay.
class.
Hmm.
Yeah, and then in there we talked about like photosynthesis.
Okay. What did you do? What did you exactly do in photosynthesis? Could you tell me that?
Wait, let me double check in for 1 second.
Yeah. Once, if you can check and tell me.
I don't think we did photosynthesis in molecular biology. So
Okay.
we did
Yeah. All right. No problem. Then because photosynthesis is there, right?
first molecules to metabolism, water,
Hmm.
carbohydrates and lipids,
Hmm.
proteins, enzymes, DNA and RNA, and then DNA replication.
We did that. Okay.
Yeah,
Next.
that's it. Oh, yeah, that's it. Up to 2.7.
Okay. 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 All right. Fine. Okay. So we'll just take a small count of photosynthesis. Okay.
Okay.
All right. Okay. Let's see. Let's see this. Okay. So this, did you understand light harvesting complex? Yes.
Yeah.
Yeah. So in photosynthesis, when we talk about process of photosynthesis, photosynthesis is divided into, let me check here. Photosynthesis is divided into light reaction and dark reaction. Light reaction and dark reaction.
Mhm.
Light reaction. Light reaction occurs in grana. Okay, grana of chloroplast. Dark reaction occurs in stroma of chloroplast. Sana, you remember the structure of chloroplast we had done, right?
Yeah.
Yes. Okay, once please go through the structure of chloroplast. Okay. Light reaction will produce ATP plus NADPH plus H plus. Clear?
Yeah.
Okay, produce ATP plus NADPH plus H plus. Okay, now these are called as these are called as assimilatory power. Clear?
Mm-hmm.
Assimilatory power. Okay, next. This assimilatory power, this is used by, this assimilatory power is used by dark reaction. Okay, used by dark reaction to produce starch. Okay,
Yeah.
see the points. See the points. What do light reaction do?
It produces ATP and
Hmm.
then
NAD plus
yeah.
H plus.
Yeah.
Dark reaction occurs in the stroma chloroplast, and whatever the products the light reaction is producing, that is used by the dark reaction to produce what? Starch.
Mm-hmm.
Okay. Light reaction will produce oxygen. Dark reaction, here it will trap carbon dioxide.
Yeah.
It will not use oxygen. It will trap carbon dioxide to finish the process of photosynthesis. Clear or not?
Yeah.
Okay. Okay. Anything to you have not understood here?
No.
Okay. All right. Okay. So photosynthesis light reaction will produce ATP and NADPH that will be used by dark reaction to produce. Okay, dark reaction. All right. To
Mm-hmm.
produce what? Starch. Yes or no?
Yeah.
Yeah. See the simple thing. Okay. Clear? Okay.
Yeah.
So let's see here. When we talk about light reaction, when we talk about light reaction, we say cyclic photosphorylation. Photophosphorylation. And also we will have non-cyclic photosphorylation. Okay, so here phosphorylation, phosphorylation, addition of phosphate group. Okay,
Mm-hmm.
so cyclic photophosphorylation, cyclic, it produces only ATP. Okay, only ATP.
Yeah.
Non-cyclic photophosphorylation, non-cyclic photophosphorylation. Okay, non-cyclic phosphorylation, this will produce ATP plus NADPH. Okay, ATP, non-cyclic photophosphorylation, ATP plus NADPH. Okay, is this clear?
Yeah.
Yes, the cyclic will produce only what?
NAD.
No, no, cyclic,
No, only
cyclic.
ATP. Only ATP.
Non-cyclic.
Non-cyclic ATP and NADP.
NADPH plus H plus. Clear?
Yeah.
Yeah. See here. All right. Okay. So
Mhm.
ultimately, what happens it is in non-cyclic photophosphorylation that the starch will be formed. Yes or no? And that is the
Yeah.
main aim of the process of what? Photosynthesis. Clear or not clear?
Clear.
Okay, cyclic photophosphorylation. So, okay, fine. Understood, Sana? Yes. Any doubt?
No.
Okay. Read this sentence. Read the sentence. Cyclic and non-cyclic. Cyclic photophosphorylation produces what?
Um. Only ATP.
Only ATP. Yes or no?
Yeah.
Yes. Non-cyclic photophosphorylation produces
ATP and NADPH.
DPH plus H plus. And this ATP and NADPH plus H plus, it will be utilized to form starch. Is this point clear? Any doubt?
Yeah. No.
Yes, any doubt here? Cyclic photophosphorylation, non-cyclic photophosphorylation. Yes, Sana?
No. No doubt.
Oh, okay. All right. So photosynthesis you have not done even little bit, right?
We've done a bit in HL.
Okay, okay, okay. Not here, right?
Yeah.
Okay. All right, no problem. These things have you done?
No. Not really.
Okay. Okay. But did you understand or not?
Yeah.
Okay. Got it? All right.
Yeah.
In cyclic photophosphorylation, only it will work only at 700 nanometer. Non-cyclic phosphorylation. Okay. Non-cyclic photophosphorylation works at 680 700 nanometer.
Mhm.
Okay, these are the important things, Sana, that you need to know about this particular concept. Clear?
Yeah.
Yeah. So, with the help of photosynthesis, the plant produces what? Starch. Yes or no?
Yeah.
Yeah. Okay. Now, see. Dark reaction. Where does dark reaction occur, Sana? What did I write here?
It only occurs in the stoma of the
Yes,
corpus.
of chloroplast, stroma of the chloroplast.
Yeah.
Yes or no? Occurs in
Yeah.
the stroma of the chloroplast. Okay, so
Mhm.
dark reactions function will be to produce what? To produce starch. Okay, dark reaction to produce what? Starch. I have written here. Understood? Yes, dark
Yeah.
reaction and light reaction. Okay,
Mhm.
so let's see here. See, non-cyclic photophosphorylation and cyclic photophosphorylation, what they are? They are light reaction or not?
Yeah.
Yes?
Yeah. Light reaction.
Okay. So, light reaction, cyclic photophosphorylation and non-cyclic photophosphorylation. Cyclic is occurring at 700, non-cycling at uh, non yeah 700 700, cyclic at 700 only, non-cyclic at 680 and 700.
Hmm.
Okay,
Yeah.
dark reaction. Now we'll see dark reaction. Dark reaction, where it will occur in the stroma. Dark reaction. Dark reaction occurs in where?
The
Occurs
stoma.
the stroma.
Mm-hmm.
Occurs in the stroma.
Yeah.
And dark reaction we have Kelvin cycle, and after that we have, oops, no, instead of giving here, Kelvin cycle and Hatch and Slack cycle. Kelvin cycle and Hatch and Slack cycle. Clear?
Mm-hmm.
Yes. Understood?
Yeah.
Okay. All right. 1 second. Okay. So dark reaction is Kelvin cycle and Hatch and Slack cycle. Kelvin cycle occurs. Kelvin cycle is also called as C3 cycle, which occurs in C3 plants. Hatch and Slack cycle. Hatch and Slack cycle is called as C4 cycle. Okay, and this takes place in C4 plants. Clear? This takes
Yeah.
place in C. Any doubt here?
No.
Okay. All right. Okay. So here, Sana, one thing that you need to remember here, we have two type of plants. How many type of plants? We have C3 plants and we have C4 plants. C3 plants, they need more carbon dioxide. Okay, more carbon dioxide. Because why? They need to trap. Okay, yeah, they need more carbon dioxide. C3 plants. C3 plants will have a cycle which is called as Kelvin cycle. Okay, Kelvin cycle. Whereas C4 plants, they need less CO2. Okay, C4 cycle, C4 plants will have a type of anatomy in their leaf which is called as Kranz anatomy. Kranz anatomy. Clear?
Mm-hmm.
Okay. And in C4 plants, they will have C4 cycle as well as C3 cycle. Both the cycle will be. You understand?
Yeah.
Yes. Why? Because it is C3 cycle that will help in the production of starch. So the C3 cycle in C4 plants will help in the production of starch. Any doubt here?
No.
Okay. Clear or not?
Yeah.
Yeah. Understood, Sana? Yes, today's class.
Yeah.
Any doubts? See, a little bit of
Mm,
photosynthesis also. I just did it. Okay. Now at least the basic you know, right?
yeah.
Okay. So you say that the third topic they have started. Yes.
Yeah.
Yes. So the third topic is genetics, right?
Yeah.
Yes. So which one of genetics they have started? Can you tell me? What is gene? What are alleles? Yes.
Yeah, we've done that. We're on like 3.3.
So
We're
sorry, 3 point.
3.3. Wait, I'll read it out really quick.
We did 3 point. Just a
So
moment.
we did
Meiosis.
first, yeah, we're on meiosis. So we first did genes, then chromosomes, now we're on meiosis.
Okay, first you did meiosis, right?
Yeah. No, first we did genes. Now we're doing
Gene,
meiosis.
meiosis. Okay, no problem because you need to know what is meiosis. So what I'll do is that in the next class, upcoming class, I will do first meiosis. Okay, then I will come up with the genes and yes.
Yeah.
All right. Okay. So So shall we end the session here, Sana?
Okay.
Okay, understood, right? So in the
Yeah.
upcoming session, first I'll do meiosis because meiosis is important. After that, you'll understand what is genes. Okay.
Yeah.
Okay. All right. So we'll end up the session here.
Yeah.
Okay. 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 Bye. Take care.
Oh, you too.
Yeah. Bye.